Hey everyone, welcome to episode 18, Remastered. Today's episode is about gene modification, and it will be the last episode in this series on genetics. This episode is also unique in the sense that it's the first episode that will discuss man-made breakthroughs in genomics and biology and how they affect the world that we live in today. Biotechnology is an emerging field with tremendous medical potential, and artificial gene modification is only a small part of it. As I'll cover in this episode, the ability to modify and manipulate genes on a molecular level has revolutionized our world to such a degree that it affects the food we eat, our medicine, and how we understand the history of life on Earth. This is actually a good place to start our episode, as the history of life has largely been revealed by the study of the changes of genes over time. Single-celled organisms like bacteria and archaea have relatively simple genomes. The larger the organism, the larger the genome tends to be. Single-celled organisms of all sizes can interact with one another through a process called lateral gene transfer. If you understand reproduction to be vertical gene transfer, like genes being passed down a family tree, then what is lateral gene transfer? Well, as you might have guessed, it's the transfer of genes between two independent mature cells. The genes are being passed side to side between lineages, as opposed to being passed down a lineage. Archaea and bacteria keep their genomes in the form of a giant loop, along with smaller loops of DNA called plasmids. These plasmids, as well as replicated chunks of the larger DNA loop, can be traded between cells, where they get incorporated into the receiving cell's genome. Evidence shows that almost all bacterial and archaeal species are affected to some degree by lateral gene transfer, and this has prompted some scientists to question the current understanding of the evolutionary branches of archaea and bacteria. Eukaryotes, like plants and animals, also have natural techniques for gene modification. All organisms, including bacteria and archaea, have things called transposable elements. These transposable elements are small snips and tidbits of DNA. They're short little sequences that can be inserted into almost any other location in the cell's genome. These elements are copied, and the copies can be stitched into a new DNA sequence, causing mostly negative, sometimes neutral, and rarely positive mutations. Transposable elements are kind of like playing biological boggle. You shake it up and you see what you get. Humans have numerous transposable elements, but many of these genes produce inactive proteins, as we lost the genes for an associated activator protein. So we produce a bunch of transposable elements, but we don't have the blueprints for the proteins that activate them. So these transposable elements create proteins that just float around in our cells doing nothing until they get broken down for parts. The eukaryotic cell's genome also carries with it strange little repeating sequences. 2 to 6 bases will make a microsatellite, or an STR for short tandem repeats, while anything longer than 6 bases is a mini-satellite. Okay, so what are these things? These tandem repeats they're sequences of the DNA with lots of particular bases in an alternating repetition, ACACAC or TGTGTG. If you consider a random DNA sequence to be like a key, then it would require an appropriate lock. The DNA sequence can only bind to a complementary strand of DNA. During meiosis, the homologous chromosomes align next to one another, with similar DNA sequences associating in a way that makes the homologs line up nicely. These tandem repeating sequences, however, can mess up this nice arrangement. Because these tandem repeating sequences are so similar, the homologous chromosomes can accidentally get them mismatched, and this puts the homologous chromosomes slightly out of alignment. During recombination, during later stages of meiosis, these misalignments in the tandem sequences can lead to tandem sequences being unequally distributed between daughter gametes. Some daughter gametes might have more tandem sequences or longer sequences, while others might have the opposite. Now, this is where this stuff starts to get really interesting, because this means that every person has a unique frequency of tandem sequences in their genome. This is like a DNA fingerprint, and it's the characteristic that's used in forensics to study DNA evidence. The various criminal justice systems of the world use the evidence gathered from DNA fingerprints to determine innocence or guilt, to exonerate those falsely imprisoned, and to identify individuals engaging in repeat offenses. Now, you might be left wondering, what about the misaligned chromosomes? Doesn't the misalignment of these tandem sequences cause the cells to have differently sized chromosomes, even within the same organism? The answer is yes, it does, and the cell's response to this problem is a major source of genetic diversity in eukaryotic organisms. When the misalignment occurs in meiosis, gene-sized segments, basically long sequences of DNA, or small chunks of the chromosome, can be entirely deleted or added. When the cell goes through this DNA to check for errors, it notices the misalignment, and produces a copy of a nearby gene to fill the gap. The end result is gene duplication, or two copies of the same gene right next to one another on the chromosome. This is pretty convenient, because if the cell needs to express these genes, a regulator complex only needs to make one pass to transcribe two copies of the protein. This has the potential to make the cell respond more strongly to the associated stimulus. For a simplified example, consider a drop in the pH. If the drop in pH is strong enough, this stimulus causes a cell to express proteins that protect it from acidic conditions. The cell with two copies of this gene would produce twice as many protective proteins as a cell that just has one copy, and thus it can protect itself better from the acidic conditions. This better protected cell is more likely to survive in the acidic environment, and it's more likely to reproduce in the future to pass on its two copies of the gene. Furthermore, with two copies, the cell can survive even if one of those copies mutates, as it'll still have one functional copy of its gene. This is where the genetic diversity comes in. If you have a duplicated gene with a mutation, that mutation can induce some effect. It can be negative, it can be neutral, or in rare cases, it can be a positive effect. Most of the time, it'll be a negative or a neutral effect, producing non-functional sequences called pseudogenes, or it'll produce a non-functional protein. But on rare occasions, the mutation will have a positive effect. The mutation might improve the protein's efficiency or its effectiveness, or it might be slightly altered in such a way that it can associate with its unaltered protein cousin to create a protein complex. As both gene copies get expressed at once, their products would easily be able to associate and form the functional complex, or an enzyme activator relationship, or something like that. In this way, groups of related genes stay together in a form-follows-function relationship. This group of genes is also called a gene family, which operates in much the same way as a taxonomic family. Genes can duplicate and create sibling genes, so to speak, and these sibling genes can experience mutations. And the slow accumulation of mutations in all of these sibling genes, these duplicated genes, will create gene families, and these can help diagram when related species diverge from a common ancestor. Another biological method of gene modification is RNA splicing, but I talked about RNA splicing in more detail in the previous episode. So right now, I'm going to transition to briefly talk about the scientific fields that are focused on studying genes and gene expression, and the equipment and the techniques that they use to acquire data. Perhaps the most general field that I want to talk about is functional genomics. Those who work in the field of functional genomics dedicate their work to studying the complex relationships between regulatory molecules, between genes, and between the proteins that they express. They seek to understand why genes are grouped or activated in the ways that they are, and how the expression of genes can be coordinated in response to an outside stimulus, or as part of the organism's natural development and maturation processes. Another field of genetics is proteomics, or the study of proteins. Where the genome is the organism's full set of genes, the proteome is the organism's full set of proteins. Proteomics studies the interactions of many or all of the proteins in an organism's body, all operating either as discrete groups or as links in a biochemical pathway, or all the proteins in an organism's body in their entirety that interact to maintain homeostasis and keep the organism alive. Both proteomics and functional genomics use a powerful tool called a microarray. A microarray is basically a flat piece of silicon substrate, studded with millions of biomolecules like proteins or single-stranded DNA sequences. The biomolecules that are studded into the substrate are known, so when scientists introduce foreign biomolecules to the microarray, they can see which known molecules react to the unknown foreign molecules. The microarray is extremely useful for seeing how genes are expressed at different ages or in response to outside stimuli, and how proteins interact with one another inside the cell. Understanding how all of these individual systems interact with each other to sustain a greater whole is the purpose of systems biology. The systems biologist studies the emergent properties of massively complex chemical systems, properties like metabolism or growth. The systems biologist seeks to unify the knowledge of functional genomics and proteomics into a cohesive whole, which would then describe the macroscopic effects of uncountable chemical reactions and trillions of cells to maintain the life of a single organism. In order to continue this kind of research, scientists need more than just the humble microarray. They would need other equipment, more sophisticated and more powerful equipment, like a PCR machine, a di-deoxy sequencer, CRISPR, or some other recombinant DNA technology. A PCR machine is a device that allows scientists to quickly print out millions of copies of a specific DNA sequence. The known DNA sequence is put into a watery solution, where the temperature is increased until all of the hydrogen bonds holding the strands of the DNA molecule together break apart, and the associated strands separate. DNA synthesis is initiated through primers, and the associated primers of the known DNA sequence get mixed into the solution. The temperature is then lowered, and the primers can bind to the single-stranded DNA. This will attract the polymerase molecules, which will synthesize a complementary strand of DNA onto each single strand. The original strand of DNA has now been doubled. It's been duplicated in its semi-conservative fashion. Another cycle of heating and cooling will double the DNA again. Another cycle, another doubling. Another cycle, another doubling, and again and again. This process is exponential, so it only takes 20 to 30 cycles to produce millions of copies of your desired DNA sequence. This technique allows us to take small, tattered fragments of DNA found in, preserved tissues or maybe young fossils from extinct species and amplify them a millionfold. Then it becomes relatively easy to isolate the sequence and study it to see how it fits into the ancestral lineage of currently living species. This exact method is how we learned about the interbreeding between Neanderthals and various groups of Homo sapiens. It's how small DNA fingerprints can be amplified and examined for forensics or criminal justice. So once the PCR machine gives you 1 billion copies of your desired sequence, the next thing you want to do is figure out what your DNA sequence actually is. For this, scientists use a process called di-deoxy sequencing, or Sanger sequencing, after the scientist who devised the method. First, a solution is prepared for DNA synthesis. This means the solution is rich in deoxyribonucleotide bases, the A, T, C, and G, all of them ready to be incorporated into a DNA strand. But it also includes biomolecules called dideoxyribonucleic acids, which lack a critical OH group necessary for DNA synthesis. Because of this, whenever a dideoxyribonucleotide is added to a strand of DNA that's currently under construction, the DNA synthesis will terminate. The enzymes can't add another nucleotide base onto the 3' carbon's OH group, because it isn't there. There is no OH group on these dideoxyribonucleotides. So when you have proteins synthesizing copies of a particular DNA sequence in this solution, you end up with a large number of copies. You end up with a large number of copies of this DNA sequence, but all of them are of different lengths. They all go so far down the DNA sequence before they've incorporated one of these dideoxyribonucleotides, and they've just been cut off. Each copy was built up using regular deoxyribonucleotides, but when a dideoxyribonucleotide was added, it terminated DNA synthesis. The key thing to understand here is that this means that the dideoxyribonucleotides expose which nucleotide base sits at which position in the DNA sequence. When you line up the DNA copies side by side according to their length, the dideoxyribonucleotides on the end of each strand will tell you what base exists at that point in the sequence. You can literally go from your smallest copy, say 3 bases long, and see the end nucleotide by studying its spectroscopic signature, or its light signature. This will tell you the nucleotide at position 3. The next copy tells you the nucleotide at sequence 4, and then sequence 5, and then 6, and so on and so forth. Sanger sequencing was one of the first techniques developed for DNA sequencing, so it's relatively labor-intensive and time-consuming. I mean, you might have gotten that impression just by listening to me describe how the mechanism works. But fortunately for biological researchers, new sequencing technologies have been developed that increase the speed and accuracy of DNA sequencing. PCR techniques can be used to amplify hundreds of different genes at once, and these can all undergo simultaneous sequencing in an effort called a massively parallel approach. Other techniques operate on the molecular level, and involve the literal pulling of a single strand of DNA through a pore. The pore has a shape that gets altered depending on the nucleotide base currently passing through it, and the sequence of responding pore shapes can be used to determine the DNA sequence itself. I think this mechanism is particularly cool. Other recombinant DNA techniques use various methods to identify a species of DNA sequence and either remove it, copy it, and/or insert it into a different gene or a different genome entirely. These isolated DNA sequences are packaged with proteins or inserted into a small plasmid for transport, where they can be literally injected into a host's cells and then incorporated into the genome. For example, this technique was used to isolate a growth hormone gene, which was then used medicinally for treating pituitary dwarfism, and it can even be used cosmetically to improve height, to improve bone density, and to increase muscle mass. This takes me to an exciting subject called CRISPR, or CRISPR, which is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. Essentially, a CRISPR is a sequence of prokaryotic DNA that can identify and cut out DNA that doesn't belong to the organism, like the DNA that gets injected by a virus, or a deleterious gene handed over during lateral gene transfer. The CRISPR molecule forms a complex with a protein called Cas, and together they form a functional gene editing group that protects the prokaryote by removing bad DNA. Scientists were able to modify the Cas protein into something called Cas9, and this CRISPR-Cas9 complex can be used to modify genomes at will. Basically, the CRISPR-Cas9 complex needs only be guided by a particular RNA, and it can cut out any part of the genome you'd like. The raw utility of this feature is indescribably exciting in the biological fields, especially in those fields that deal with treating genetic diseases. Although CRISPR isn't a perfect panacea. Recent studies have found that it's somewhat messy. It can make cuts and breaks and deletions where you don't want them to be, and it can lead to massively deformed genomes that are punctured with huge unintended deletions. But the science is being refined, and at the time of this recording, there's a new Cas protein called Cas12 that is supposed to be much more accurate, so we'll see where this goes in the future. Now, with respect to the medical utility in humans, it's kind of tricky. Gene therapy is a tricky science. Once an individual has identified a problem gene within their genome, like the genes that cause Huntington's disease or cystic fibrosis, Gene therapy seeks to counteract the damaging genes with healthy genes. The trick to this science involves figuring out a way to get healthy alleles of a gene or genes into cells with unhealthy alleles. This must be done in such a way that only the desired tissues get affected, and they're only affected with the right concentration, the right dose of the alleles, and this dose occurs at the right time in the organism's development. Furthermore, the gene therapy has to find a way to silence the unhealthy alleles, so that only the healthy alleles are expressed, or at the very least, the healthy alleles get expressed much more often than the unhealthy alleles. Problems with any of these variables can cause a host of potentially serious and sometimes fatal complications. Genes can be inserted into cells through mechanisms that work kind of like a virus. The virus injects the DNA and a minimal host of regulatory proteins into a cell, such that the host cell's enzymes will incorporate that DNA and begin expressing it. These techniques often involve the use of retroviruses, which catalyze the insertion of the viral DNA into the chromosomes of the host cell. Gene therapy can be used to treat a disease called SCID, or Severe Combined Immunodeficiency. Basically, there's a gene on the X chromosome that gets mutated to a point where it no longer works, and this effectively neuters the individual's immune system. The gene codes for a receptor on the T cell, which is a really important cell type in the immune system, and it renders the T cell non-functional. The individual has non-functional T cells, so even a minor infection can be potentially fatal. For the longest time, treatment was limited to keeping these kids in a sterile room for their entire lives, like a bubble. The whole bubble boy thing came from treating these kids with SCID. To address this issue, researchers took 10 male infants with SCID and extracted the stem cells on their bone marrow that code for T cells. A retrovirus was used to put a healthy, functional version of the gene into the genome of the stem cell, and the stem cells were then placed back into their infant hosts. Of the 10 infants with SCID who underwent this gene therapy treatment, 5 of them went on to grow up with normal T cell behavior and normal immune function, having been effectively cured of SCID and saved from a horrible life of perpetual quarantine. However, 4 of the children would develop cancers due to uncontrolled T cell production, and 1 of them would die from it. The 10th child failed to respond to the initial treatment, which is rather curious, but he was treated later in his life with donor cells and has since lived a normal life. As you can see from this example, gene therapy is not a clean-cut science. It's not perfect. What works for many people may not work for others, and could even lead to fatal complications in a select few. The technological sophistication of our gene therapies has to be improved to minimize dangerous errors like the ones that cause cancers later in life. We need to think about how gene therapies can be used not only to treat, but to permanently cure and put an end to genetic diseases that have plagued families for generations. This opens the door for cosmetic and elective gene modification, but I'll get to that at the end of the episode. It goes without saying that gene modification isn't limited to medicines. If you eat food, chances are you've eaten genetically modified food, or you've eaten food that's been produced by genetically modified organisms, also called GMOs. Now, when I say this, I don't mean organisms that are modified slowly through artificial selection over thousands of years. I'm talking about organisms who've had their genes radically modified overnight, in labs, by people sitting hunched over their desks looking into microscopes. This kind of gene modification has revolutionized our world by providing food that lasts longer, handles temperature extremes better, and grows larger and brighter, with better yields, and some would say with a better taste, although I suppose that's a matter of personal opinion. Probably the most famous GMO is called golden rice, and this is kind of ironic for a reason I'll get to in a few minutes. Let me explain the context for you. Half of the world eats rice as a staple food, as an integral part of their diet. However, rice doesn't provide the full complement of nutrients that a human body needs. For example, rice doesn't have vitamin A. Its leaf and stem tissue produce a protein called beta-carotene, which gets modified into vitamin A in the human body. But the actual rice that you eat, the actual carbohydrate-dense little rice grains, those don't have beta-carotene, so your body has no way to derive any vitamin A from the rice. This can be seriously problematic, as vitamin A deficiency can lead to blindness. Tragically, near 250,000 children in Southeast Asia develop blindness every year because of a vitamin A deficient diet. This is clearly a problem. So what's the solution? Scientists wanted to see if they could get beta-carotene to get expressed in the rice tissue itself. First, they identified a protein in the rice that can be modified into beta-carotene with the help of just 3 enzymes. The genes that code for these enzymes were isolated and then strung together with endosperm regulatory sequences. These would ensure that the genes are expressed in the endosperm, which is the fancy word for the tissue that eventually becomes the grain of rice. The researchers then identified a bacterium with a plasmid capable of injecting its own DNA sequences into the DNA of a host plant. They used this bacterial plasmid as a vehicle to deliver the enzyme genes into a rice embryo. As the embryo grew into a plant and produced rice, the researchers found that much of the rice was a rich golden color. As the rice endosperm grew, the endosperm regulatory sequences made sure that the genes were activated. This produced the enzymes necessary to turn the natural precursor molecule into beta-carotene. The product rice was packed with beta-carotene, giving them their rich yellow color and the name golden rice. As the human body makes vitamin A out of beta-carotene, the golden rice effectively solves the vitamin A deficiency problem. What's kind of darkly ironic and really sad is that despite the apparent benefits, golden rice has not been approved for commercial production and sale in any country. However, despite the fact that golden rice is kind of being held on legal hiatus, so to speak, rice is hardly the only genetically modified plant that's out there. Potatoes, tobacco, alfalfa, maize, papaya, corn, cotton, tomatoes, canola, eggplant, apples, bananas, squash, soybeans, and carrots are just a handful of the total number of modified plant species. When scientists perform some kind of genetic modification, they typically try and make the species more durable and efficient from an agricultural context. This means that they find ways to repel insects and other herbivores that would damage the crops. They find ways of minimizing the impact of weeds, and they find ways of improving the nutritional content and of improving crop yields. For example, the first GMO crop approved for sale in America was a strain of tomatoes that had the name Flavor Saver. The Flavor Saver tomato had a longer shelf life, as the people who engineered it were able to disable the expression of an enzyme that eats away structural proteins in the cell walls. Ordinarily, this gene would be expressed, and it would cause the tomato to eventually get soft and mushy, get softer and mushier as it aged. But by suppressing this gene, the researchers allowed the tomato to stay firm and edible for significantly longer periods of time. This also makes it easier to ship the tomatoes, as they last longer in transit. Another GMO crop is the Arctic apple. These were modified to silence a gene expressing polyphenol oxidase, which is responsible for the browning of fruit tissue when it's exposed to the air. As a result, Arctic apples take a lot longer to brown from oxygen exposure than other apples. Then you have Cavendish bananas, which have been modified to express nearly 10 times as much vitamin A as an unmodified banana. There's drought-guard maize, which is a species of corn that can endure prolonged periods of drought, where it has little to no access to water. Other modifications can make these various agricultural plants tolerant to various herbicides and pesticides. If the crop plants that you're trying to grow can be made immune to a particularly strong herbicide, then that herbicide can be used to kill weeds without harming the crop species itself. Similarly, powerful insecticides can be used to kill parasites and insectoid herbivores without harming the crop plants. Crops like potatoes, squash, zucchini, and cucumber have all been modified to be resistant to various viruses. Now, agricultural plants aren't the only genetically modified organisms out there. Many animal species have also been modified to suit our agricultural needs. Researchers in China, Argentina, and New Zealand have all independently engineered cows that express human proteins in their milk. These cows have been effectively engineered to produce human breast milk instead of natural cow milk. A lineage of pigs, called enviropigs, were engineered to break down phosphorus compounds in their food, leading to a huge reduction in the amount of phosphorus that they excreted. This is really important for ecosystem health, as the waste from thousands of pigs all shitting in the mud on an industrial agricultural farm creates runoff that's rich in phosphorus. When this waste phosphorus gets into water sources, it acts as food for all sorts of algae species. When the algae eats the phosphorus, they also end up consuming a lot of oxygen, which can create dead zones in the water where there's no oxygen for fish and other aquatic organisms. So by modifying pigs to reduce the amount of phosphorus they excrete, we can minimize algal blooms in nearby bodies of water and better protect fish populations. Now, even the fish themselves are being modified. There's a company called Aqua Bounty that patented a GMO salmon that they claim can be raised to maturity in half the time as a normal salmon. Besides agriculture, animals are also modified for scientific purposes. Fruit flies, sea anemones, and zebrafish are model organisms that are routinely used in genetic modification experiments. There's a famous study using fruit flies where the researchers were able to activate the genes for an eyeball in the cells of a fly's leg. Basically, they were able to grow a fly's eyeball coming out of its kneecap just by messing around with regulatory sequences. A fluorescent protein found in jellyfish has been transplanted into pigs and cats, which literally makes some of the mammal's tissue glow with a soft fluorescent light. These fluorescent proteins are extremely useful because they can be tracked and monitored as they move through the body. This way, they can illuminate the pathways and responses that cells make during transplants, or when they're exposed to certain diseases. Some hoofed mammals, like sheep, pigs, and goats, are modified so that their organs are much more acceptable to human hosts. This is a really important medical purpose, as animal organs with these modifications are much less likely to be rejected by the body. Should you get into a car accident and need an organ transplant, these genetically modified pigs and sheep and goats all have organs that your body is much more likely to accept. From food to medicine, genetic modification can be used to create a much more sustainable world. GM crops can give more yields, they can grow more sustainably, and they can feed more people. Genetically modified animals can provide novel sources of nutrition, new research opportunities, and even organs that are better suited for transplant. With some of the technologies that I discussed, we can potentially start modifying our own genomes, although modifying the human genome, especially the germline, opens the door for very important ethical discussion. Exactly how far should we go with our own genetic modification? What's the limit on what's acceptable? For example, as a matter of personal freedom, should we let people modify their somatic cells however they desire? I could see this being more or less acceptable, because somatic cells aren't going to be passed on to their offspring. Somebody can perform as many somatic cell alterations as they want to really morph and alter their body as much as they can, but when they have children, because they didn't alter their germ cells, when they have children, their children will just be normal, healthy humans with none of the weird genetic doodads that their parents have added to themselves. But the ethical discussion gets a lot darker when you start to talk about germ cells, and whether or not people should even be able to modify their germ cells, because germ cell modifications are going to be passed on to their descendants. They're basically modifying the genome of any offspring that they choose to have. So the obvious question is it ethical to give birth to a child with gene alterations that they did not consent to having? In the case of a genetic disease, you might think that it's unethical not to cure it. You might even argue that once they've been made aware of their heritable genetic diseases, people should be compelled to get gene modification to their germ cells. So if there's any argument for germ cell modification, the strongest basis would be to argue for removing genetic diseases, stuff that would cause Huntington's, cystic fibrosis, celiac disease, and sickle cell anemia. The ethical discussion gets even darker when you start talking about cosmetic modifications. How far should personal freedom go when you're talking about performing cosmetic or elective modifications to the germline? I mean, it's fine for someone to choose to modify their genes to make their eyes blue, for example, but that would be somatic cell alteration. But what if that modification gets put into the germline so that it gets passed on to the person's children, and their children are guaranteed to have blue eyes, or whatever modification it is that they made? I mean, you might think, well, blue eyes aren't that bad, who cares? But what if it's more extreme than that? What if the individual wants to change their skin color, their limbs, their genitals, or their facial structure on a genetic level? We're not talking about just switching from one to the other as if we were RPG characters. We're talking about drastically altering them to mutate the morphology and change the physical structure, just to kind of experiment with our own human biology as a weird substrate of, personal freedom almost. It's a really ethically weird place to be, and there's a lot of biological danger here. One of the most important things to keep in mind when you're talking about germ cell modification is that whatever modifications you make to the germline, even if it's some weird, goofy cosmetic thing that somebody wants for whatever reason, that change, because it's in the germline, will persist in the human lineage forever. As long as their descendants continue to reproduce and have children, that artificially modified gene will persist in the human lineage, and we will quite literally be taking evolution into our own hands, for better or for worse. And I don't know about you, but I don't think our species is very good at long-term planning. And I am really hesitant and really worried about people who are eager to start altering the germline and to start trying to control human evolution. I think that we'll be playing with forces that are much larger than we can comprehend, or much larger at least than we think, and will create some really serious problems that might last for a very long time. As the technology surrounding gene modification becomes more and more sophisticated, we'll have to have these conversations. We'll have to face these ethical issues, and we'll have to address how to responsibly handle our species' genome for the sake of all future generations. Well, I think I can end the episode on that happy note. I hope this episode helped you to shine a little light into the world of genetic modification. This is a really important science, and it's becoming increasingly important and increasingly common in our technologically dependent world. So I think it's really important that you understand at least the basics of modern gene modification. I hope you give a little thought to ethical questions surrounding gene modification, and the potential places that gene modification can take us into the future. And as always, thanks for listening.